The prostate is a small gland located beneath the bladder that is involved in the male reproductive system and functions to produce fluid that nourishes and protects sperm in the semen. It consists of an epithelial and a stromal compartment which incur significant changes during the initiation and progression of prostate cancer. Prostate cancer is the most commonly diagnosed male cancer, with over 16,000 new cases and 3,000 fatalities expected in 2017 in Australia alone. It is estimated that 1 in 7 men will be diagnosed with prostate cancer by his 85th birthday, and in the same period, 1 in 30 men will die from it. For this reason, our company have put years of research into finding a new and suitable therapeutic treatment. Our drug candidate will target one of the underlying principles that govern tumorigenesis, deregulating cellular energetics. As a result, it will also target the following hallmarks of cancer: sustaining proliferative signaling and resisting cell death. Cancer cells reprogram energy metabolism to ensure survival and proliferate. One method used to achieve this is to increase glutamine use for the biosynthesis of precursors such as NADPH, GSH, proteins, nucleotides, antioxidants, and important amino acids. The importance of glutamine in cancer has been highlighted by research showing that glutamine metabolism restriction is effective in suppressing cancer growth by sensitizing HeLa cells to FAS ligand, TNF-alpha, and lead to shock-mediated apoptosis. Glutaminase is an enzyme highly expressed in rapidly growing tumour cells and functions to convert glutamine into glutamate.
In cancer cells, glucose becomes redirected from the mitochondria and glucose is converted into lactate. Therefore, glutamine serves as an alternate carbon source for the intermediates of the tricarboxylic acid cycle. To regulate this, cancer cells increase the expression of glutaminolysis through the action of the enzyme glutaminase 1. This converts glutamine into glutamate. Glutamate breaks down into ATP as intermediates of the TCA cycle, use this energy to support and sustain proliferation. GLS1 is the first enzyme to initiate glutamine metabolism. At the dimeric surface of the active site, a non-competitive antagonist will bind to the kidney GA isoform that is expressed in the prostate, forming an inactive tetramer, and will initiate a conformational change of the key loop of GLS1 blocking glutamine from binding. Thus, this will selectively inhibit GLS1 enzyme, preventing glutamine metabolism and decreases ATP levels. This triggers apoptosis and there will be no energy supplied to the cancer cells. MINC oncogenes upregulate glutamine metabolism by increasing the expression of GLS1 to compensate for the energy loss and to maintain the integrity of the TCA cycle. From this, MINC is considered to be sensitive to glutamine depletion, thus inhibiting GLS1 and will rapidly shut down the MINC oncogenes. P53 becomes mutated in prostate cancer, and eliminating the primary function enables cancers to survive high levels of DNA damage and leads to rapidly growing tumours. Mutated P53 inhibits the GLS2 enzyme, as GLS2 can act as a tumour suppressor as it can decrease the level of reactive oxygen species in cells. RAS keeps fueling glutaminolysis through the overexpression of glutamine dehydrogenase. The RAS kinase can regulate the intrinsic activity of GLS through phosphorylation in the cancer cells. Through the inhibition of GLS1, this will create a delay in the G1 to S phase cycle transition and will ultimately lead to DNA replication arrest. This cell cycle delay results in a decreased activity of the RAS oncogenes. This proves that RAS cells are dependent on glutamine and glutaminase.
As you can see here, studies have shown that GLS1 expression is much higher in prostate cancer tissues, PCA, compared with benign prostate hyperplasia, BPH tissues. This study proves the clinical significance of GLS1 in prostate cancer and consequently how our drug, which inhibits GLS1, will be key in overcoming prostate cancer. Another great study completed just last year highlights how GLS1 can be inhibited using BPTES, which, like our drug, is a non-competitive inhibitor of glutaminase. As you can see, the results of the study show how BPTES can ultimately result in a decreased level of cell proliferation in pancreatic cell lines. We want to use your funding to improve our drug, which elicits the same results but in pancreatic cancerous cells.
Prostate cancer is a major source of financial burden and sickness among Australians. Thus, we believe our drug, which acts as an inhibitor, is an efficient method that will starve the cancer of nutrients, leading to cellular death, and thus warrants funding for further development. With this grant, we will strive to create an orally administered drug, which is preferential route of administration as it is less invasive than other treatments, such as surgery and chemotherapy. We aim to maximize its pharmacodynamic properties and maximizing bioavailability, efficient and overall absorbency to create an optimal treatment. Through this, We believe we can improve the patient's quality of life and life expectancy and allow them to be cancer-free.